I'm going to show you how to process resting state functional MRI data using CONN. So this is lining up with Module 9 for the class. And this is a data set that is publicly available, but we've limited it to 10 control participants and 10 participants who have brain tumors. And we're going to be able to do this analysis. We'll do a very simple analysis. And in this case, the data set is compressed in a zip file and it is within blue CLP 7934 share module 9. Leave it there. Don't move it. Don't do anything in this folder. All we're going to do is copy this out of this folder into your own directory. So I like to open in terminal. So I'll click on open in terminal. And so I am now in this module 9 folder. Type ls. There's only one zip file. If there's anything else in there, somebody else has created an extra file in there that should not be in there. And the person did create it, then delete it. Don't delete this zip file though. So copy this in. So what I'm going to do is I'm going to copy this into my folder in share. But the first thing I'm going to do is actually I'm going to make a module 9 directory. And there's multiple ways to do it. I'm actually going to make it from where I am. So I'm going to make a directory, not in this current one. Again, don't do anything in this current directory. I'm going to make it up, // is up one level. And then into my directory, the slash, and then I'm going to call this module 9. So make dir, and I'm not making it where I am, I'm making it up one level and then down into my directory, and then it's going to be called module 9. So that is going to be the output target for this, for copying this file. So I'm going to copy and hit tab to autocomplete. I'm going to copy it up one level and then into module 9 within my directory. So again, copy it before you do anything. It shouldn't take very long. And now I can cd into my directory, module 9. So do all that with yours. And so now I have this zip file here. So now I can just unzip it. And this won't take very long to do, so you can just do it here on this login terminal. Is okay. So we have 10 participants who are control participants, and we have 10 who are patient. So unzip, we can see we have a JSON file for the T1, a Nifty file for the T1, resting state bold, the blood oxygen level dependent scan, that is a functional scan. And so we have each of those for everybody. So this is a BIDS format file, and there were some other files that were created. So now I'm going to go into that location. Sorry, I already went into that location. I'm going to copy that location, what I meant to say. And I'm going to go into that location over here on this window. I just wanted to show you. So you can do it this way, where you can put in the path, copy and paste, copy the path from the other window, paste it in here, and we'll be right in that location. So now that has extracted into a directory called resting state fMRI class, named a little bit different than the zip file, and that's okay. That's what we expect. And so we can go in here and have all the participants. So now what I'm going to do before we get started is go into the derivatives file, and I'm going to create a new directory called CONN. This gave me an error, which happens regularly, so I can refresh this, or I can make another directory within here, but within derivatives, within the resting state fMRI class, derivatives, I'm going to make it, call it CONN, and so now that directory has been made. So I can refresh this and we should have this CONN directory. That's going to be where some outputs are saved. And so the class, we have all these files, and the ones we want to pay attention to is this participants file. So download this file. This is a tab separated value file. So download it and open Excel. I've already downloaded it, so I'm going to open that now. And so I downloaded it. I just right-clicked on it, open Excel, and here's the file. So this could be a data set, meaning it is a data set, but this could be any data set, one that you have from a research mentor or whatever the case may be. But this is the participant ID, and there are the 20 participants. The IDs match our the data we have. We have sex, we have age, tumor type, so our control group, and our patient group, who all in this case have grade 1 meningiomas. There's tumor size, all sorts of different data. So our analysis for this class, for this homework, is going to be a between group analysis. Is there a difference in connectivity between those who have brain tumors or had brain tumors, those without? And but we could do a lot of analyses. We could do something just within our patient group. Is there a correlation between tumor size and functional connectivity? Is there a correlation between smoking status, caffeine intake, alcohol use, all sorts of different things? Now, there's data within here that we want to pay attention to, because this is going to be important for processing the data. So this data set actually very nicely has this fMRI TR. That is a time to response, and this is something that we're going to use. So we'll come back to this, but this is in milliseconds. So this would be put in kind of 2.1 or 2.4 seconds, but these are milliseconds, and this is something about the scan itself that we will need to put into the pre-processing, and we'll show you in a little bit. So we'll come back to that, and that's important. Those are data that you can get from the scan, from the JSON file itself. So for example, I'm going to go into CONN1, into functional. Here's the JSON file. I'll view it. There is something called repetition time, or TR. It's often put backwards. Time to repetition. Repetition time is a 2.1. But in this data set, in this file, we had CONN5 had a TR of 2400. So we'll look at that. CONN5 had we'll look at the JSON file, and they have a repetition time of 2.4. That's seconds. Within this other file, this is in milliseconds. But these are going to go into the setup for when we do pre-processing very shortly. Okay, so that's something to be aware of with functional data. It's a little bit more complex than structural data that we've dealt with so far. This functional data, there's this thing called that repetition time that will influence it. There's slice timing things that will influence it. This gets to be a lot more complex and just very basic. We're just doing a very basic introduction. If you do this for research, there's a lot more you're going to need to know. But for our purposes, again, this is just dipping your toes into this big ocean of functional MRI data processing. So those are the data. So what we're going to do is open up a MATLAB session. So the MATLAB session, you can come over here, Interactive Apps, MATLAB. Now this will have a different setup. We won't need this much. But what I like to do is I like to use a version of MATLAB that's a little bit older than the newest one. Sometimes the newest one just can have some problems with processing, whatever. So I'm going to use 2020B. You could use something newer. Any of these ones should work, but I generally don't recommend using the newest version. The other thing to keep in mind is if you're doing a study, you want to use the same version for that entire study. So you want to keep that. And that's why there are some older versions on here, but 2020B should be fine. So let's request 2 cores and 16 gigabytes of RAM. You can use the class account for this. 24 hours. You want to make sure it looks like that. 2 cores, 16 gigabytes of RAM, class account. Okay. So I'm going to launch this now, and this will take a little bit of time to start. It could be anywhere from seconds to hours, just depending on how busy HyperGator is. You're requesting burst access, so under the QoS it was COP7934-B, and we're doing that because although we're only requesting 2 cores, we don't have enough cores for the class allocation for everybody to do this at the same time. So as to be courteous, we want to make sure you're using burst allocation, which is lower priority, but gives us enough cores for everything. So I'm going to pause the video and then come back once this has started, which could be anywhere from a few more seconds to hours from now. Okay, we are ready to go. So I'm going to launch MATLAB. And the first thing I'm going to do is I'm going to change location. So this is on my home drive, which is on HyperGator. You have one as well. Don't do anything in that location. So change location, blue CLP 7934, share, and then I'm going to go into my directory. And so that just kind of gets us in the general location where we need to be. If for some reason you can't see that, you can try going over to Applications and CD blue CLP 7934 share, and then your directory. You can try doing it over there first and then typing it in there. You shouldn't have any problems, but in case you do, try that first and then try typing that again. So to use CONN, which is a software we're going to use, we need to have 2 other software available. We need to have MATLAB available, and that's what this is here. And then we also need to have something called SPM12 available. That's in addition to CONN. So we need to come over here and set the path to tell MATLAB where SPM12 and where CONN are. And so I already have this set up with some kind of various kind of a setting. So I'm going to add CONN on here. I actually already have CONN in a different location than SPM12. So I have SPM12 in 2 locations. You should have an SPM12 already because we did Cat12 processing, this VBM analysis, that was already done. We used MATLAB. And so you should have that. But if not, we can add the folder. And where that is on blue CLP7934 share neuro tools. So blue CLP7934 share and then neuro tools. Oops, I didn't actually want to quite add that. Guess I'll go this way. Blue, share, NeuroTools. Okay, so here's SPM12 and CONN. So SPM12, I'm actually going to remove this NeuroTools. So SPM12, and then we also want to add in the NeuroTools CONN. I double clicked on it, which is okay. You don't have to double click on it, but we would just want to have CONN located right there. And so we're setting the path to the software so MATLAB will know what to use. So you hit save And it might have a pop-up saying, I can't save in this location, or do you want to save pathdef.m file in some other location? Go ahead and do that. Save it in your blue CLP7934 shared personal directory. Don't save it in the module directory or somewhere else. You can save it there. And then close it. So you just want to make sure CONN and SPM12 are showing up in there. So then you should be able to type CONN all in lowercase. If it gives you an error, then that means you didn't source the path correctly. You didn't save it. You need to do that last step again. So this will open up CONN, should be version 22.8. And I'm going to make this window bigger so I can do kind of this full screen with it. So it's starting. It takes a little bit of time to start up. So now we are going to create a new project. So I can just click New. And I'm going to save this within Module 9, that resting state fMRI class, derivatives CONN. Now I'm just going to keep this file name. I like to be a little bit more descriptive, but this is fine. We'll keep it like that. And I'll put save. So I'm putting that within derivatives CONN, again within my directory. So now this is where we do the very basic setup. So essentially we're going to go down this column and get the pre-processing done. So how many subjects do we have? We have 20. And each one should only have one session, one run, meaning we just have one scan for each person. We have two scans. We have a structural scan and we have a functional scan, but one functional scan per person. So I'm going to put 20, and then right here is this repetition time. So now if you remember from here, we had this fMRI TR. So this is very nice to have in this data set. And this would be what I recommend if you have a data set. Pull that TR out. Hopefully it's the same for every participant. This one is not, which is a little messy, but that it's here. But what we're going to do is I'm going to copy this, and then I'm actually just going to create a new tab down here. I'm going to paste it in here because I want this in seconds. So how do I get this in seconds? I can just divide this number by 1,000 and just drag it down. And so then I can copy that, right click, or in my case Command C, and then I'm going to paste it over in here. And or you can type it in one by one. That runs the risk of being a little bit problematic. But I'm going to come over here and clear it. Now, there's a weird thing that this says it will kind of can override this. So actually I need to go over and copy this again just to make sure that it's there. So I copy it, then I can paste it in the clipboard. And then now that it's in the clipboard, I can come over into this window. Oh, I want to delete that. Delete it first over here out of this repetition time, and it'll make it easier. And so then I have the numbers in there. It doesn't matter if they're not side by side. It'll work this way. And then I can right click. You're not letting me right click, but I should just be able to at this point paste it. And I'm using a Mac, but to get it to paste in here, the keyboard shortcut is actually a Control V. So if you're using Windows, it's a Control V, not the command. But even if you're using a Mac, still pasting in here is a Control V and not the command. And so it matches sort of the copy and paste from the Windows side. And so then that gets it in there, or you can type them in there one by one. But so the repetition time, it would be a lot easier if it was the same for everyone. That's the ideal, but this data set, that's not the case. And don't worry about what this means. This is just something about the scan that goes into the pre-processing. Okay, so that's in there. There should be 20 of them. So now we need to load in our structural and our functional scans. So structural scans, these are going to be the anatomy ones, the T1 weighted. So module 9, resting state fMRI class. Okay, this is going to be where we want to search. So we can come over here to files, and I'm going to search for a T1 weighted .nifty.gz. So I type that in with the asterisks. I do find, and it's going to find 20 files. If it doesn't find 20 files, there's some problem with your data set. Start over. You either had an incomplete extraction or you had double extraction or something. And so I'm going to come over here now. I'm going to select all 20. So shift click, select all 20. And then I'm going to come down here, same sort of thing. Select all 20 files, 20 subjects, 20 files, first and last. So it's control 01 last patient 17, which is what we'd expect. And then I click import and it will load it. So what it's going to do is it will assign each one in order. So the first one to subject 1, second one to subject 2, on down, and it will uncompress them. So these are .gz files. SPM just likes .nii files. So that's pretty fast to do. I click OK. So then I like to just check and make sure that it loaded. Ideally, you would go through each participant and check, but we'll just do a subset to show that we were getting a different scan for each person. They're in a different slice of that, get a location and all sorts of things. Now we do the same thing for functional. And I'm going to select all 20 right away. Then we're going to come over here, and this functional scan is now called bold.nifty.gz. So I type that in, I click find, and it will find 20 files. Again, if it doesn't find 20 files, you're searching in the wrong location, you've extracted too many, you've extracted too few, something's wrong. Probably want to start over with copying the file, the zip file over and extracting it again. 20 files, import it. That'll just take a little bit of time. This will take a little bit longer than the structural. These are bigger volumes. This is a functional scan, so it's taking a picture, effectively taking a picture of the brain over and over and over again for a period of time. And so a lot bigger file. So I'm going to pause the video and then come back, but this doesn't take too long to do, but it can take some minutes. And that just depends on how fast the computer is. In this case, Hypergate is quite fast, but again, it will take a little bit of time. So we'll come back when this is done. Okay, this is now said files are assigned, so I'll click on the OK, and we can move on. So again, as before, I just want to show that the scans got assigned to each participant and that they look a little bit different from person to person, as they should. People's heads are shaped differently, and if we notice, these are not nearly as detailed as the structural scan. Resolution is lower, and that's because of the sequence. The functional scan generally are going to have a lower resolution, but it looks a little different because we're focused on patterns of blood flow and not so much the structure. So the scans are here, and now I'm just going to show you these. You don't have to click through the rest of these until we get down to pre-processing, but ROIs is where if you wanted to look at a particular atlas, you could load one in here, a region of interest, and this is going to be very specific to a research study, research question, more advanced. There are a couple of built-in some network, these atlases, these are regions of interest that we can use to look at connectivity between these regions. and a couple of different ways to look at it. One's very structural, another one's a little bit more of a functional atlas, meaning that these are areas of the brain that are working together at rest or in when you're doing a task, and all of that is more than we need to cover in this processing tutorial. But that is there, and you could add in new regions of interest in there. Won't worry about this. I just want to show you that you can see the scans, but you could load in other scans as needed or pre-processed. Again, don't do anything in there. These conditions, if we had repeat scans for everybody, we could set up multiple sessions and give them names and all sorts of setups in here. If we were doing a task, so somebody was in the scanner and they were watching a video and responding to it, that's where we could set all of this up, but we're not going to worry about that. I'm going to skip first level covariates. Second level covariates, we're going to come back to this in a later video once we do the pre-processing, but this is where you can put in those other values. So this is where we could put in age and sex, what group people belong to, tumor size, whatever we want to put in there. This is where your behavioral observation, these variables, anything can go in there. And so groups, but we'll come back to this one. And then options, we're not going to change anything in there. Those are more advanced. So we're going to come to pre-processing. We're going to keep this pretty simple. This is not necessarily the best way to pre-process functional data. It's not wrong. It's well accepted. But to keep this simple, we're just going to do the default processing pipeline. So go there, click OK. And that puts in several steps that are run. And this is a good way to process data. There's one more step we could add that would improve this. That's a little bit more advanced, so don't worry about it. And but this is the pre-processing, and we're going to make a couple of changes. So before we get to this, we want to come over here to Tools, the HPC Options, Configuration. Now, hopefully you have an HPC Slurm profile. If you don't, you can create a new profile, or you can take null profile. Sorry, you need to come here and do new and try it out. But we want one called HPC Slurm. If you don't have it, then you need to create it, do new, and then have it match what we have in the canvas for this. On the canvas for the module for the class, there is it kind of has what should go in there. So you should have something that looks like this. Again, there's text in there. You can copy and paste. If you don't have this, you can create this profile. But hopefully you have it. But the only thing I'm going to really check for is in here, we want to make sure that we're requesting enough time. And this is separate from the time we requested previously. This is each job gets submitted. We want to have enough time and RAM in particular. So that should be there. And then class account here. So this is defaulting to use mine, which I could use for processing, but I'll switch this over to the class account. And so what this will do is we're going to process brains, and we're going to process as many at a time as we can. So we want to make sure we're requesting enough time, enough RAM, and we're going to request the class account with burst access. So save that. And I'm going to save it for me. And that should be set. Now, when the class access ends at the end of the semester, I'm going to have to go back and change this back to my account and make little tweaks. But this is, make sure this profile is there, and that within this in file section you have 20 hours, 8 gigabytes of RAM, and then the class with burst access. So then I can just close that window. And now we're ready to do the pre-processing. So we don't want to do local processing run on this computer. We want to do distributed processing run on HPC Slurm. We want to process all data, all subjects, all sessions. We only have one session, but we want to process all subjects. So now we have 20 participants, and so ideally we would request 20 cores for this. So number of parallel jobs, we will put in 20. Now we should have, we might not have enough resources for if everybody in the class was doing this all at the same time, but we're going to guess that not all of you are going to be doing this at exactly the same time, and so we should be okay to request 20 jobs. And so what that's going to do is it's going to use one core for each person. This will get through the processing much faster than it otherwise would. So we're going to split this into 20 jobs. And there's a couple other things about data. This again gets to be a little bit more advanced, so we're just going to provide, I'm going to tell you what this is. So these data were acquired on Siemens scanners. And so Siemens scanners acquire slices of the brain. Again, don't worry about this, but slices of the brain, they do it in this interleaved manner like this. And so our slice order is, you can just click on interleaved Siemens. Other scanners use a different process. This can be very complex. So ideally, you know this, but if you don't, you might have to get in touch with the physicist to put together the sequence for the study. It gets to be a little bit complex. Interleaved, or you can pull it in from BIDS format, which would probably be the best thing to do, but which we should be able to do on this data set because it is in BIDS format. However, I know this is Siemens data. It was acquired interleaved, so we're going to pick interleaved at Siemens. So I'll put OK. And then this next pop-up, outlier detection. We want to generally be pretty conservative. And what this means is anybody, so from slice to slice, or from, I should say, sequence. So a bold sequence, these functional MRI is like a movie of the brain. And so this would be like from frame to frame. If somebody moves more than 0.5 millimeters, then we're going to identify that as an outlier. And that's generally about as high as we would go, meaning somebody might do, 0.9 millimeters, but that's quite generous with motion detection. You might need to do that depending on your sample, but generally you want to use this 0.5 millimeters as your setting. So use conservative settings, push OK. This is fine, we can just do OK. Smoothing of 8 is fine. Sometimes you might want to smooth at 6. And what that means is it's just a little bit more detailed, where you're going to see these little activation spots in the brain. But 8 is a good default, and so we're going to leave that. But again, what you pick in here can depend on what research has been done, what others have used before. But 8 is a good default. We'll push OK. Now it's going to start going. And so this is submitting the jobs. And I'm just going to give it a little bit of time to make sure that it gets started. And then I'm just going to let it run. I'm going to pause. We'll come back once it's done. But hopefully, if you've done all the setup correctly and you've made those changes as needed within that tools, HPC options, configuration, hopefully that this should start to work. So here's a little window. We'll click this log history. So it says submitted. So this will give you some information about scans. If you click over on the main MATLAB window, you can see that. So what it's done is it's submitted 20 jobs. It's created 20 little submission scripts, so those SBatch shell. It did this all automatically, handled it, and it said that they are now running. And there they are. They are running. So one thing I do like to do is click on Advanced Options. Let's see if that'll, there we go. Advanced Options, and now we can see up here that these different jobs are running. Why it's not letting me quite see everything. I think it likes this window size. Normally it doesn't show these issues. But sometimes you can, I don't know why, again, why it's doing this right now, but you should be able to go in there and see some information about these, the jobs, and see updates. But don't worry about it. It should be working. You might get, if you're doing this, you might have some of these end with errors. A lot of things can go wrong. We're just going to assume things don't go wrong. If you're following along and you've done everything, you were kind of fastidious, very conscientious and careful with all of this, it should work. We shouldn't have any errors. If you're getting errors, it's very possible that there were typos or you did something not quite right on it. That's the learning process. But there are other things that will happen if you're using a different data set or whatever. There are a lot of things that can cause problems in here and start to troubleshoot, and it can take a while to troubleshoot, but hopefully everything goes smoothly, and this will take time. I can't estimate. We'll just say this is going to take an hour to finish. It could be a couple hours. It could be many hours for this to finish. It just depends on the processing. So again, be patient, and we'll see you in a while. One comment I want to make about this is you can close this window. Let me show you that. So don't delete this, that's running. You want to keep that running, but you can close that window. I can come back, I can refresh this. Again, I can close down the computer, I can go somewhere else, whatever is happening. But I can come back and I can go to my interactive sessions and I go, oh, okay, here's my MATLAB session. It's been going. And again, I'm going to check this in a while, an hour or so. And I can go launch MATLAB. Everything will come right back up to where it was. And so this is still running. So again, just leave it. But again, you don't have to leave this window up if you don't want to. Don't worry about shutting down your computer. This is running on Hypergator. So you can go ahead and close that and come back to this later. And so that's that first step. And so we'll come back with the next video in the step of this next step of the processing. The first step of pre-processing finished up. It didn't take that long to do. And so I'm going to now show moving on from there. And what I did do is I closed the window that was showing the kind of the processing, and this again didn't take very long, maybe 30 minutes tops to do that first step. And so I closed that window and I got a little notification that said the job is finished, would you like to merge the jobs? And so essentially what it does is it split it all up into 20 different jobs, and then you want to merge them all back. And so those are done. Said here that happened, it finished and everything. And the other thing I do want to recommend is regularly saving the project. It does save when you do steps, but just kind of get in the habit of project save, and that will help prevent any issues that might happen if something happened and this crashed. Okay, so the first step of pre-processing finished, but we're not done with pre-processing, so we want to do the next step. So the next step is we're going to skip the QA plots. Normally you would do that, but we will not do that. We see I got this little disconnection that happens occasionally, and so I'll refresh the page. launch MATLAB again and go back in. Hopefully it doesn't keep disconnecting a lot. It doesn't matter, it's still running, it's just I'm losing my connection to it. And so I'm going to skip the QA plots. Again, in a real analysis, you would do that. You would look at the data more in depth. But we're going to click Done. And in this case, this is going to run the pipeline. It did some initial setup for it. But we're going to do this distributed run on HPC Slurm and do start. And then I want to do 20 parallel jobs again. Push OK. And then it's going to start running. So this will submit and it will go, and it will take a little while to run, but once this is done, we'll come back. This is running, but I did want to show one thing. If you look in this job manager, and I was able to make it a little bit larger, and you can go and click refresh, and it was just stuck on submitted, but I can do refresh and it will show that these are running. You can also see whether or not they're running in some of the other windows. So this says submitted, which doesn't really help us that much, getting that log file. But if we go back to the main window here for MATLAB, again at the top, it is showing that the jobs are actually running, not just submitted. And so again, the information is shown in here that can be useful to be aware of. So this job manager, I'm just clicking over to get all of these back on here. I just want to show something. This job manager can be closed. And so I can just exit out like that. And it looks like it's going to move on, but it says no pending jobs. And so I can click on that and click on advanced. And then I'm going to make this larger. And so this is opening that window back up. So this is running, and some of the jobs may take less time to run. So they might say stopping or completed. If any of them have an error, then that's going to be an issue and it takes some troubleshooting. Hopefully you don't have any of those issues. If you are getting it, again, it might be because of an error in setting up the data, that you just did something wrong, and that's part of a learning process. And it might be easier just to kind of start over at that point, and rather than sit there and try to figure out why it's not working. But if you've followed everything in the video, it should be working just fine. And this stuff, that's not going to be true for every data set. But you can kind of refresh this, and we can see one is finishing up already. This isn't taking very long to do. But what I want to show is you can click on any of these and we can see logs, and it shows all the steps. What it's doing segments the data, imports information, denoising, and doing all of this, and is finishing up. And we can click between the different nodes, and so essentially you can check and see where each job is. So this job is on step 5 of 7. This one is on step 4 of 7. That one's on step 4 of 7. And that one is just about done, 5 of 7. Again, you can sort of use this to kind of check and see where the data are, but this will take a little bit of time. So I saw one of them is finishing up really fast for whatever reason. The others are going to take a little bit more time, but we'll pause the video and come back. And this step is not taking a long time. Larger data sets can take much longer to do, especially if you are having to run multiple participants on each core. Here we requested 1 core per participant, so it's pretty fast to do. But again, it can take some hours for this process. And if you have enough data, it can be days for this process. So be patient, and we'll be back in a little bit. This finished up and didn't take particularly long to do. So what you can do is you can check, you can refresh and see, okay, this is finished. And let's say you had closed this window, which is fine. Up here, there may or may not be a little thing that says pending jobs. If not, you can come over to Tools, HPC Options, Active Pending Jobs, and it says no pending jobs associated with this project. And so this it was done, and but I closed all of that, and let me show you sort of what happens. So that's closed, and it didn't really didn't let me quite finish this, meaning that I needed to put everything together. And what it's done is not much. So we can look at this, and it said finished, everything done, save the project, no pending job associated with this project. But the problem is that now we can't go on to the next step. So we should have a step when this one is done. So again, we had done pre-processing, and then I hit click done, and we're doing that processing. We should be able to move on to this denoising step. And that hasn't appeared. So let's do this. We're going to reprocess, but we're not going to recompute. We're going to skip already processed subjects and ROIs. And why I'm showing this is because, again, just depending on what's going on, you might click on something, something might not show up as being done. And so I'm going to start this. We'll request 20 parallel jobs. And this actually won't take very long at all, because it's going to go, oh, these have already been processed. We're going to just move on. So I'm going to click Show Advanced Options. Make this window bigger, and this will show that it is submitted. So now this first part will take a little bit of time, because it can take time for the jobs to actually start. But we're just going to show you if you needed to reprocess, or let's say you came back and you added data later, which I'm not doing for the homework, so don't worry about it. But if you're doing this for a real thing, you could go and add in data in the setup for the project, add new participants, add them at the end, and do all of that. Sometimes I just start over and reprocess everything. It can be a little bit simpler. It just depends on where you are. Like how far you are, how much you have to add, or whatever the case may be. But you can add participants. But for showing this is submitted, the jobs are still waiting in line. You can come over and check. Still submitted. And again, this is going to skip anything that's already processed. So it's not going to take very long, because we don't have a lot of data. There's only 20 participants, and they've already been processed, so it's not going to take long. But I will pause it, and we'll continue on. So you may not have had this happen. You may have been able to just move on, close and continue, and do all of that. But I just want to show you, if you had closed all of this and had X'd out of here, you might want to go through this process again. But you can also just wait a little bit and see if the denoising showed up, which it actually is now, and which is okay. But we're just going to let this wait. So really what I wanted to say is, if I had given this more time before going and let's reprocess this, but I'm not really reprocessing anything, then the denoising step, like, it would have shown up. It was still doing some background processing, even though it said it was done. So just kind of give it some extra minutes before moving on. But again, you can reprocess, or if you want to just check and make sure, oh yeah, did this really finish correctly, then you can go and do what I did and reprocess. But in this case, I'm not, as I said, not really reprocessing anything, because I told it to skip things that were already done. And so again, I was just showing you this because you can go back and deal with the data later, and add new data, or whatever the case may be. So let's let this finish up. So I'll sit here a little bit and then we'll click refresh. We'll see if it's done. And now it says finished. And we can watch in the background. It's loading, it's doing some things, it's flashing, it's looking like it's anything that's saved. So now I'm going to click finish. And it is moved on to this denoising step. So again, that whole little process you might not have to do, that kind of skip already processed and opening all that up again. I just want to show you in case that was an issue when you couldn't move on to this denoising step. You might need to do your setup and click done again. You might need to go into tools and look for active or pending jobs, things like that. And again, so if you had done the pre-processing and you had done and you can't move on, either just wait a little bit, a few minutes to see if you can go on to the denoising step. If not, again, click on done, do the reprocessing again, but skip already processed participants and try it, and then you should be able to move on. If not, then there's some other issue, and at that point you might want to start over. So the denoising step, so all MRI data is very, very noisy. And one of the and one of the goals with all the processing is we want to find that little bit of signal in all of the noise. Structural data is very noisy. Functional data is even more noisy. And that noise comes just from being in the scanner, meaning like literally noisy, but that's not really what we're talking about. We're talking about this is tracking, in this case, we're tracking hemoglobin in the brain and looking at changes in the oxygen level of hemoglobin, and that's a really small signal that's happening. And that the interplay between the protons, these molecules, protons, all of this happening in the brain and sending off this little signal that the RF receiver is picking up. This is just a very noisy process. Well, there's a lot of noise, and there's also noise from just the blood flow itself, heartbeat, breathing, all sorts of things that are in there. And so looking at the denoising, one of the things we want to look at, how do we know if the data are processed, pre-processed decently? You can do some quality assurance plots. We're not going to worry about that. We're not going to look at those right now. But one of the things we can look at is we want to look at our before and after. So if you have a pretty good denoising, this connectivity values distribution, we'll get to that later. But again, we're looking at just functional connectivity of the brain. This is an R value, correlation value between every spot in the brain and every other spot in the brain. That this should shift down to center around 0 and be pretty normal afterward. And so this is before and after. And so after looks good. I know that's subjective, but it's pretty normal, centered around 0. It doesn't have to be perfectly centered around 0, but ideally it should be. The other thing is we can look at this line right here, and that should be pretty close to 0. You're always going to get a little tail over on the end, but otherwise, again, say the before and after. And then the other thing we can look at is down here, and there's some banding in there and everything. It should be pretty smooth, fairly homogeneous afterward. And this global signal should be pretty flat afterward. A little bit subjective. There's some other ways to kind of turn this into a metric and be more formal about it, but that's just a visual thing. So ideally, you would go through and look at each participant before and after. Is it looking pretty smooth afterward, fairly normal, centered around 0? And so most of these are looking pretty good. Again, I didn't click through all of them, but these are this is a publicly available data set, and so the researchers have already done a little bit of cleaning of the data, meaning they weren't necessarily including participants' data that is going to process this, do this pre-processing. That'd be really bad, but it is possible. So you know, you have to go through and check everything. You just go through one by one, but all of these have looked pretty good so far, except for this one. So this one's a little bit weird because it started out fairly normally distributed and then it shifted over. So this got nice and smooth, the line's very smooth, but for subject 13, it kind of did something odd. And so what that means is we should probably go and we'd go back and look at this participant. So okay, subject 13, let's go look at him. We'll go back to setup. I'm going to go to functional. Actually, let's look at structural first. So subject 13, this looks okay. So now this has gotten normalized to standard space, but what do we see? We see this person has a very large tumor. That person has a tumor as well. But subject 13 has an enormous tumor. And we can see that over here. I saw this file open. Here we go, looking at the tumor size in centimeters. This is actually subject 13, so this is row 14, but this is subject 13, has a 78 cubic centimeter tumor, which is massive. And so that is kind of throwing off the processing, actually. So we can look at the functional data for this person as well. And this is after processing. And there's the tumor. It's still showing up on here. So what we're going to do is we're going to keep this participant in. Now, whether or not we should do that is a little bit more of a technical decision, and it's more advanced than this tutorial. But that's part of why we go through and we look at the denoising steps and everything. And then ideally, like this person again, subject 13 here, this distribution got worse than it was before after all the denoising. Like there's just, this is just kind of very different compared to everybody else. Yeah, meaning like it's completely red and it shouldn't be completely red. This person actually did quite well before and after. And kind of flattened out. So 15 again is also sort of weird. And so there's actually some challenges with this data set, but we're using it because it's sort of an interesting one. These are brain tumors, meaning participants who have brain tumors, and so there's the issue of the brain tumor there in the data, and that throws things off. And we're just going to assume that these are all good to use, which is not necessarily the case, but for our purposes, that'll be it. So we go through and the denoising, where we say, okay, the denoising looks good. Well, if you decide, okay, these really aren't good, we need to exclude 2 participants. We definitely need to exclude participant 13 and maybe 15, but we'll say 13. You could go back to setup. I could go back in here to number of subjects. I could change it to 19 and go here, and it said, who do you want to remove? And I could say subject 13, and then that would remove it and we could move on. But I'm going to cancel and we're going to just keep everybody in. But we shouldn't necessarily keep everybody in because of how the things are being processed. But this is for class. I'm not trying to publish this. Okay, so we looked at this. We're saying, okay, we're good to go. So then you go to done, and you make that bigger, run on HPC Slurm. We can recompute for everyone. We don't need to recompute because we actually haven't run it, but we can just leave it as the default. And again, because this step we haven't clicked done yet, but I'm going to do start. We're going to split this up into 20 jobs and hit OK. And it'll go. This will actually be really fast. And I might pause the video, but because it could take a little bit of time for this to actually start. But this is a fast step, fast processing. And so we'll let this go, and I'll come back in just a minute or so. But again, you can always click on advanced. I like to make this a little bit bigger, and you can refresh occasionally just to see if it has started yet, but we'll be back in just a little bit. This is almost done, and so I've refreshed this. We can see finished, 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 and then there's one that was still finishing, and so this will be done very shortly. So what we've done so far is we have added 20 structural scans, 20 functional scans. We've done some initial pre-processing, which in that case is skull stripping the structural scan. It is moving structural scan into a standard space, kind of a template brain, forces everyone's heads to go in the same space, essentially, the brain. And it does some smoothing of the functional scan and some other pre-processing. Again, we're not going to cover exactly what it's doing. This is just first pass at getting your feet wet with this. And then we're doing this denoising step, and this one should be about done now, finished up. So now I click the finish button, and so that means we'd looked at it, it had kind of applied all of this denoising, which is just smoothing out some of the other noise that happens in the scans. And now we're ready to move on to analyses. There are different types of analyses you can do. There are what we call a seed-based connectivity. So that is starting somewhere in the brain and connecting to all sorts of other places in the brain. And so we can look from regions of interest, or we can look from one area of the brain, one region, to everywhere else in the brain. And so that's kind of a basic one that is a nice thing to do. And so we're going to do done in this case. So we just highlight that, we just click done. And let's also run this HPC Slurm. Going to start. And this should be quite fast to do. 20 jobs. And this will get them all submitted. It can take a little bit of time for them to start. Again, these are using the class burst access. And so that is a lower priority. So it is possible for them to take seconds, minutes, even hours to start. So again, conceivably, this could take a while to happen. If for whatever reason your MATLAB session ended before everything was done processing, which would be okay, as long as you have saved the project. You can go back in, start a new MATLAB session, load the project, and it would be where you were. So everything was already done. And so that's one of the benefits of this, is a lot of this is, again, just kind of save it regularly. I haven't been saving it formally, but it does get saved in a lot of the steps. If we look over in here, saved this project. And so now all 20 jobs are running, but I do like to save it manually, but it, each step it does do some saving. So if you're saving regularly, you can come back and let's say your MATLAB job completely ended. And by ending, I don't mean the window just closed, meaning like this job stopped and you had to start over again, that doesn't mean you need to start over from the beginning. You can go in, do a new MATLAB job, launch it, and you would go in to MATLAB and you could open up CONN again, kind of like we showed in the earlier in the walkthrough, and you go in, and then instead of doing a new project, you open up the project that you already have. So I won't show that, but that would be the process if you needed to go back. So some jobs over here have already finished. So again, this is a pretty quick step. But if you're over here on the CONN side of things, and that's where, if needed, you can go back and load the project in later. But save it regularly and you should be good to go. So I will pause the video. This is just going to finish up another minute or two, but we will come back. Okay, that is finished. Now, okay, what are we ready to do? We're actually ready to do some analyses. However, what analyses do we want to do? We want to do a between group analysis. So at this point, we're actually going to go back now. So the data are processed, and I could go back, and I can go back to this first level analyses. We can look at these. So each subject, we can go in and click in and look at processing. So this is part of the default mode network, medial prefrontal cortex. So here, and this is looking at connectivity to other parts of the brain. So these would just be for each individual kind of raw correlation values. And so this is one thing you can do to check the data. So in here, we should see a hot spot, this orange, red, yellow. So medial prefrontal, you should see another one back here, and then two more back here on the side of the brain, the parietal lobe. So you can kind of do that and go through and see if we see that general organization. Now this person, it's a little messy. We don't kind of see that clear organization, but here we go. And so actually subject 3 does look different. That might be real. It also might mean that something was weird about their processing of the data. As we can see, there's actually possibly something wrong, because if we look, time series is nice and variable for this person, nice and variable for that person. I mean, it should be variable, blood flowing in and out. But subject 3, very low over here. And so something happened, and we should do more quality assurance. We should be looking at all sorts of things. There's a lot more to do here. This is just, again, a very basic introduction. So going through this tutorial doesn't mean you're ready to go off and do this on research data and publish. There's a lot more to do, but this will get you kind of this first pass. But we look at these, and one I did want to look at is that subject 13. So this is the person who had the tumor, and we've got the same threshold. And what we're seeing is we're actually not seeing really clear organization at that same threshold, meaning we're not seeing any signal really show up in the brain, and which is abnormal. And what that means is that the data, this person probably should be excluded. I'm not going to worry about that for the homework. But again, this is a little bit messy data, and we're keeping it a little messy because if you happen to be working with people who have brain tumors and are interested in this, these are going to be issues. A lot of other data sets are going to be a little bit easier to deal with, but need to be aware of potential issues. But we should see some general organization of these, and meaning correlation values and all of this. Okay, but let's just pretend that everything processed exactly as expected. So how do we do a between group analysis? So this is where our second level covariates come in. So all the pre-processing is done, well, at least for our purposes. But what we have is we have some various covariates we can put in, and we can look at these. So valid scans, we actually have this person who has 1 valid scan, this person who has 13, that person who has 55, this person who has 45, and those are outliers. They should have closer here, like 180, 178. They should be around 180 or so, or it's close. We're going to lose some. But one rule is anybody who is missing at least, who doesn't have at least, say, 5 minutes of scan. How do you know 5 minutes of scan time? Well, that's for that repetition time. So remember, we had either 2.4 seconds or 2.1 seconds. And so you can actually do a quick calculation. And so this person right here, this person has 45 valid scans. So 45 times, and that was participant 3. So participant 3 had a 2.1 TR. I'm pulling that in from this data set. Participant 3 had 45 valid scans, and so times 2.1, that means that person had 95 seconds of scan time, meaning their brain, we're looking at blood flow of their brain for 95 seconds. That's not enough time to get really any clear picture versus these other individuals who were up close to 180. That disconnect again. Again, over here, 164, 142. We'll look at this first. So here's for somebody with 117 right here. And so 117, that was participant 1. Oh, we're getting into this regular disconnecting. That was participant 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So participant 10 here had that 2.4 second. So 117 times 2.4. You don't have to do all this calculation. I'm just showing this. So they had 280 minutes of scan time. Seconds, sorry, which is 4.68 minutes. And so technically, this person also doesn't quite meet a cutoff. So about 5 minutes is what we would want left. Now, we could be a little more generous and say we'll leave only 4 seconds left, 4 minutes left, or we could do it and say if they have at least 50% of their scans left, because again, if they're getting, meaning this is getting identified as an outlier if they've moved more than 0.5 millimeter, and so this means that these people are moving too much essentially during the scan. And so what we really should do is go in and exclude participants. Looking at these, we're gonna go back and do that now. And so who do we exclude? We exclude participant 3. We're going to keep this person in. Technically we could exclude them, but we'll take out participant 11. So 3, 11, 13, and 15. That means we're losing 3 of our participants who have tumors, and we're losing 1 of our controls. So we're going to have 9 and 7. So 3, 11, 13, and 15. So we're 3, 11, okay, so we're getting rid of 4 participants. So we are going to be a little bit more robust. Said we're just going to leave everyone in. We'll not leave everybody in. So change this to 16. Okay, so 3 needs to be gone. And we want to get rid of 11, 13, and 15. So 3, and I'm going to use control, maybe 3, maybe this is a command. Oh, you can double click on it. So 3 is gone. 11 should be gone. 13 and 15. Oh wait, no, that's actually getting rid of them. 3. I'll tell you what, we're going to do it this way. I'm going to do it one by one. Normally I should be able to control click, and it's not letting me. So I'm gonna delete from the bottom. So I'm gonna do 19. So I'm gonna get rid of 15 first. So let's get rid of 15. Okay, so we're gonna get rid of participant 15. So now I'm gonna go back, let's change it. Let's now do 18. And I think it was still thinking it has to do a little bit of setup. And it is still working on this. So this take a little bit of time. So this is a bit of a process. While that's thinking, I'm going to pause this and we'll come back once it'll let me move on to deleting the next person. It has to do a lot of like moving data around and kind of sorting this out, so it'll take a little bit of time. Okay, removing that participant took a few minutes, and so this is going to be a little bit of a process, but remember we took out subject 15, still need to take out 13, 11, and 3. And so we need to get down to 16. I'm going to try one more time just to see if it will let me remove these. So no, these have been reordered, and by reordered just meaning who was subject 20 before is now subject 19. And so we need to go back, and so we already got rid of subject 15. This is why I'm going from the top down. And so subject 13 would be the next person, and that person has not been reordered. Let me see if I can do multiple clicks on this. If not, then I'll have to do one by one. 13 and 11. This is not letting me click more than one at a time. I don't want to remove. It's not letting me do more than one. You might be able to do more than one at a time, which would be a lot more efficient. But so 18, I'm going to take out participant 13 now. Again, this is going to take a little bit of time to do. There probably took about 5 minutes for it to do everything. You can come over here and see steps. See if this is actually doing anything. There, we're getting this back in the main MATLAB window. It's saying busy. So we need to wait for that to not be saying busy anymore. Those will take a little bit of time. So again, I'm going to keep doing this back and forth. So if you want for the homework, you can leave in those 4 participants. I wouldn't really recommend it because it's really going to throw off the data. But these are fake, these are real data, but this is just a practice analysis, so it doesn't really matter. But this would be kind of the process of taking them out. Sometimes what I would do is I would just go, okay, those 4 were bad. I'm just going to start over, process the 16. I would like delete everything. I would go in and delete them out of the data set itself in my copy of it and just say, well, I'm not going to use these ones. And then I would just go and I wouldn't have to worry about this. But this is the other way to do it that's a little less destructive. But again, it didn't take that long to kind of get through this, so could do it, but that's sort of the process. It's a little bit more involved than some of our other things we've done. Okay, removing that participant actually didn't take quite as long this time. And so okay, this is done. It's not saying busy anymore. This came up. So I'm going to go in and we'll take out participant 11 now. So I'm going to go down to 17. Push enter return. I want to take out subject 11. Again, this only works if you're doing it from the subject 20 on down. If you're going the other way, you're going to have to change who is who because everything will kind of move up one subject. And so subject 11, hit OK. I will take them out. Again, this is going to take a little bit of time. It's not, see, it's showing busy right now, so this takes a little bit of time. And then once that's done, then we'll take out the last person and keep going. That participant removed is now the last one. This is participant 3. We're going to remove. And select yes, remove. And this should be done pretty shortly. Then we will finish up. This is done. We've removed those 4 participants, and we shouldn't have to do anything else because we had already done all the other steps. And again, they're not going to show up here in this list. So before, one of the problematic ones was subject 11, and we had just a solid red, and these distributions looked weird. Again, technical term, weird, but this is not that participant. They have now been removed from the data. Now again, you don't have to remove those 4 participants, but and that would save you some time on this assignment, but just for completeness, I showed you removing them because they really didn't need to be in the data set because they were too we just didn't have enough data left of the scan. In particular, one of them, we really had no data left. But I want to show you, so now we want to get other data in. So if we look at this valid scans, again, here we have a kind of a good range here. And we're looking a lot better than before. Before we had that one participant only had 1 valid scan, which is completely invalid to use. You can look at the number of invalid scans and all of these other metrics. Some of these can be used as covariates in analyses, but what we want to do is we want to add a few variables to this. So we want to add age, because we're going to use that as a covariate, and then we want to add group at a minimum. We could also add sex. So where do we get these? Well, we can get these from that participant's TSV file. So I highlighted, these are the ones we removed, so I'm actually going to delete these from this file, because they're not in the data set anymore. And age, so now I can just copy and paste this in. So I'm going to copy age, and we're going to add right here, add a new covariate. So this is age. So values, again, I could type them in, but that's prone to error. So we're going to clear this. I'm going to paste the age values in. Again, it doesn't matter that they're listed in a column. And then over here on a Mac, so it's not letting me right click to do it, so I can do Control V, again, even though this is a Mac, and Control V to paste them in. So now I've added age as a covariate. And we can see the range right there. So we could do an analysis, we could do a correlation between age and functional connectivity, but we want to do a group comparison. So we do have a group variable here. So, but the group is non and meningioma. So let's add a new group. And so let's add in our new column in here. Let me just paste those because Excel sometimes is a little bit too smart, meaning it's like, oh, you have copied cells. So we're gonna have a group. I'm gonna have a group 0 for none. Again, I don't have to. I don't have to do this one by one. I could fill it and kind of drag down. And so that's a group status 0 and 1. So now 1 if they have tumor, 0 if not. But let's actually call this, so this group, and I'm going to do two other things. I'm actually going to insert two more columns. And really I would only have to do one, but just kind of, let's do it this way. So this one I'm going to call, this is our tumor group, and we're going to call one control group. So what does tumor group look like? Tumor group is going to have a 1 if it is in the tumor group. So that's actually the same thing as group. So that's why we don't really need to copy it, but I'm just doing it this way. And what is control group? It's the opposite. So I could do a little if-then statement, but I'm just going to type it in like that because there's not very many. And so those are group variables, and you really want to set it up. So you set it up where one group has a 1, and then you have another variable where the other group has a 1, and kind of this vice versa. So now we're going to add those group variables. So I'm going to add these 3 variables. Okay. So down here, just click. I can just do group, and then I clear out these values, clear them from there. I'm going to copy in group. I can paste them in over here, paste them in over there. There's our group variable. And then again, technically I don't have to do anything. I don't have to do another group, this other tumor group one, because it's the exact same as group, but just to be a little bit more complete, I'm going to do it. And I have a reason for that is I could use group as a covariate, or I can use tumor group versus control group as kind of variables of interest for testing. And again, I could just use the other group variable, but this just gives me a little bit more clarity in what the variables are being called. So again, tumor group is the same, and then now we have control group. And this will just be the opposite of the tumor group. So I copy that in here, and then I can paste in the values. Okay, so we're going to keep this pretty simple. We're just going to do a between group analysis. And there's a lot of different analyses we can do, but we have this set up. So before getting anywhere, I'm going to save this. And we're ready to go over here to results. So these are our second level analyses. So these are really where we're doing our statistical analyses. So what do we want to do? We want to compare connectivity between our control group and our tumor group. And we're going to covary for age. So how do we do that? We can select all subjects. We want to use all subjects. We want to use age. We want to use all of these. For some reason, my keyboard, it's not letting me select this. So what I want to do is we're going to have to play around a little bit with this. So normally I can click and do a control click, but for whatever reason this wasn't working. And so what I can do is I can select everything, and I want to take group out of here. So how do I take group out of here? I can try it this way. Let's see if that'll work. That is not working. Sorry, this is having some technical challenges. I think some of this might be related to my keyboard, which is new. So I'm actually going to move group down. Right click. And oops, I want to move selected one down. These other ones, that's fine. I kind of moved them around. That's okay. I'm just going to move group out of the way. Hopefully you don't have that error. If you do, then this is one way to get around it. So then I'm going to go back to all analyses. And let's do a seed to voxel. I'm going to do this. So what do we want to actually analyze? So we want to analyze the effects of whether or not you have like tumor or not, if that affects the connectivity. So how do we test that? So we want to make a prediction. So the prediction in this case is if you have a tumor, you're going to have lower functional connectivity. So how do I test that? And that could be, maybe it's higher, but we can test it like this. So 1, all subjects, we keep that there. A 0 means I am covariating for it. So, but in this case, I want tumor group to be negative and the control group to be positive. Why is that? That's saying the control group I'm expecting to have higher connectivity. So that's a positive correlation. Kind of this positive, the t-test, if you will, in this case. But not really a t-test, this is an ANCOVA, and we are controlling for age. So I can push return. I can also click here and I could say, right here, difference tumor group higher than control group, or control group higher than tumor group. Sorry, I forgot to make the all subjects a 0 on it. And so this is one of the nice things, or I could look at the effective age, and that would be covariating for the group membership. But in this case, we want to say control group higher than tumor group. And so then they have that relationship there. So zeros for the other variables, 1 for the control group, -1 for the tumor group. And you can kind of do those contrasts by hand if needed. So what are we interested in? So what part of the brain should we expect there to be a difference? For simplicity, let's just say there's going to be a difference in the default mode network, which is a network that's particularly active at rest. And we're going to go from part of it called the PCC, the posterior cingulate cortex, and this will connect out. We're doing seed to voxel. This is connecting out from the PCC to the rest of the brain. So we are specifically asking right here, does the average connectivity differ between tumor group and control group subjects after controlling for the influence of age? I mean, we're controlling for all subjects, that doesn't really matter, but controlling for age. So does the average connectivity from the PCC to the rest of the brain differ between the groups? And we're specifically testing if the control group is higher than the tumor group. I mean, really it's going to test both directions, but okay, so there is our analysis. So this is a group comparison. So we can compute results. It won't take very long at all. Even with a larger data set, this doesn't take very long to do. So all the time is with the pre-processing. So we compute our results. This will be done soon. And we'll be able to look to see if there are any differences, anything that pops up. Okay, so nothing showed up. So if there were any group differences, we would see some splotches on here. Now we only have 16 people, not very many in each group now. 9 in the control group and only 7 in the tumor group. So what does that mean? That means our statistics need to be less stringent. And so this is kind of a stringent statistic. So what we can do is I can customize them, and I will get rid of, I am not going to correct for cluster size, and I'm going to bump this up higher. Now this really isn't, this isn't publishable, probably. I mean, it potentially could be. I'm not saying this is, I mean, this is not, but you could have comparable size groups and you could potentially publish data, but it would have to be very specialized question, very preliminary pilot data. I mean, this is, if anything, this is the type of data that you would use to argue for a grant to get more data to be able to see if there's anything there. But for our purposes, we're seeing these few spots. So anything that's red is, we'll want to look at these directions. So I can click on this button, and this will show you kind of the groups. So here we go. So again, this is connecting from this kind of back part of the brain right in the middle to the rest of the brain. And so we have one cluster here where our control group does have higher connectivity. Our tumor group has this lower connectivity. But lower connectivity just means lower. So the tumor group actually has stronger connectivity. It's just negative. And so it's a little confusing, and you have to be careful with some of these interpretation of results, because closer to 0 is less connectivity. And so you can actually have a really strong negative connectivity, negative correlation between areas. But this is an area, and I don't know which blotch it is. Well, I can click on it. But that's one where our tumor group actually has a more negative, so it's lower, but it's actually a stronger connectivity. And so it's negative, and but closer to 0.2 on average versus this less than 0.1 for our control group. But it's different pattern. This next cluster, our control group, has higher connectivity. It's more positive, but it is also closer to 0.2 versus here less than negative 0.1, closer to 0. And then our tumor group here has a positive, and our control group now has a negative connectivity. And so we can kind of click on those and see, or we can come over here and click on each of these and see. So here's one. So we have our 2 red ones right here, and we have a blue one. And if we go back over to that window, so this one right here, so our posterior cingulate, this is connecting out to sort of somatosensory supramarginal gyrus area of the brain, and which is actually part of default mode network. And so we're actually seeing that within the tumor group, although this is sort of just a little bit outside our kind of default mode. Anyway, don't worry about it if you don't know all these terms. But I'm just saying, so this is an area where our control group has negative correlation, negative connectivity. Control group has positive connectivity. So that's why that was blue, meaning our hypothesis that our control group had higher connectivity than the tumor group. And that would show up in red. If it's blue, then it means tumor group has higher than the control group. But in this case, again, higher means like, doesn't mean absolute value higher. It just means that our control group is negative and the tumor group is positive. And although the magnitude of the of the correlations are similar, although the tumor group is a little bit higher, meaning this is greater than 0.1, it's about a 0.7 or so, and on the negative side we're at about a 0.15. So slightly higher positive correlation versus our negative correlation. So the magnitude is a little bit different. But so the interpreted meaning, our control group has lower connectivity than our tumor group, but lower just means it's closer to 0 or negative in this case, kind of like here. And so it doesn't always mean the same thing, which is each spot. So here, for example, this is 10, negative 10, 24, 52, negative 10, 24, 52. This is an area where going up to superior and middle frontal gyrus, where our tumor group has a pretty strong negative correlation and the control group has sort of a weak positive correlation. And so maybe that's normal, meaning it should be kind of this weak positive correlation, but our tumor group is having this negative correlation, meaning when blood is flowing into that area, it tends to not be flowing into the posterior cingulate cortex. But technically the tumor group has a stronger connectivity with that area. It's just more negative. So again, confusing, and we've had to sit down sometimes with results and just make sure that we're really interpreting them correctly. And so here's a way to look at it. So for homework, what you can do something like this, and then you could get a pretty picture to display. You could either do this screen would be sufficient, or you can open up a different image like this. It'll just take a few more seconds, and you can make this window bigger and kind of rotate this around. And look, there's that one cluster right there. If I want to look at all of them, I can highlight all of them, or I can click off and open this window again, and that will show all of these little clusters show up. So this is visualization. So for homework, and you could do like that, take a screenshot, submit this, and you could be done. Or you can, there's other ways to get screenshots out from here, but for our purposes, it should be sufficient and just kind of show that you're getting results. If you want to do some other analysis, there's a lot of other analyses that you can do. And again, we could say maybe we just want to look at the effects of age. We don't want to do between group. We want to look at a different part of the brain. We want to do regions of interest, which is a little bit different. Anyway, all sorts of things, but you can kind of play around with this if you want. Just kind of get some sort of screenshot results, and that should be it.